Good morning from Stanford. My name is Drew Endy, and the talk right now will be an introduction to synthetic biology. The goals for the talk, from my perspective, are, if you've never heard about it before, to get a sense of what synthetic biology is, and even if you have heard about it, to think a little bit about why synthetic biology, whatever it is happening. So what is synthetic biology and why is it happening? The talk for this morning has three parts in the title. Meant to be a little bit playful since it's early in the day here in Palo Alto: aliens, computers, and engineering biology. So we'll see if we get through those three parts. Please very much send in any questions. We'll look to take them, and I'll definitely stop before the end of the hour so we can get to as many questions as possible. So please contribute your questions. We'll be looking forward to that. Without further ado, let's get into it. So I wanted to start with a sort of playful example around how synthetic biology is being used to understand natural biological systems. And the way to start this is an interesting paper from Francis Crick back in 1972, where with Leslie Orgel, he wrote about the idea of what's called directed panspermia. How did life arrive on Earth? And did it form here spontaneously, or was it sent here? And directed panspermia is the idea that life was sent to Earth. And so it's a very interesting paper where, for example, in figure 1, they look at the age of the universe, the origins of life on Earth, and the gap between when Earth-like planets were first expected to exist in the universe long, long time ago and the origin of life on our planet. And that's about an 8 billion year gap, which means there was plenty of time, apparently, for life to arise somewhere else in the universe, and then perhaps it was sent on its way. And if you see in the top right of this slide, Crick and Orgel are speculating on what a spaceship would look like for sending life throughout the universe. It's not a spaceship carrying astronauts, it's a spaceship carrying microbes and maybe the phages that prey on microbes and so on. And in the bottom right, just grabbing a snapshot from their paper, what's very interesting, among other things to think about, is if you have a long time to travel, if you have billions of years to travel, you could go quite a distance at a relatively reasonable velocity. So for example, if your spaceship went 60,000 miles an hour, but you had 1 million years, well, geez, you could cover quite a bit of distance. So that's directed panspermia. A couple years later, 7 years later, in 1979, another paper was published. And something happened between 1972 and 1979, namely Sanger and others got sequencing working. And the first DNA genome sequenced was a bacteriophage named Phyx174, and that was published around 1977. So 2 years after that sequence comes out, here's an interesting paper from Japan wherein they ask, is the genome sequence, bacteriophage Phyx174 DNA, a message from an extraterrestrial intelligence? So that's an interesting title. Is this a crazy paper, a good paper? Well, meanwhile, today we know there's quite a number of phage on Earth. There's about 10 to the 31 phage on the planet. That makes them probably the most numerate reproducing biological systems on Earth. So if you were going to send a message and you wanted to be all over the place, maybe you would think about putting it in a phage. Let's unpack this paper from 1979 a little bit more. When the Phyx174 genome came out, it had interesting features. So on the left here is a map of the genome. It's a circular genome under 6,000 bases long. And you can see in the top left quadrant from 9 p.m. to midnight, there's a box for gene A. That's a gene encoding a DNA replication protein. However, right between 11 o'clock and midnight, you can see another box inset within the gene A coding sequence, that's gene B, and that encodes a totally different peptide, a scaffold protein helping the particle assemble. And the two proteins are sharing the same genetic real estate in that region from 11 p.m. to midnight via basically using two different coding frames within the genetic sequence. When this came out, it's kind of interesting, right? The one gene, one enzyme hypothesis from way back was not so obviously correct in this context. What's going on here? Moreover, when the researchers from Japan looked at the details of the sequence of gene B, which you can see on the right in figure 2 from their paper, they noticed that the number of codons in gene B was 121, exactly 121 codons. That turns out to be the square of 11, the square of a prime number. Maybe that's where the aliens put their message, so we could identify it by having this puzzling, hyperdense genetic architecture. How could evolution get overlapping genes like this? So what the Japanese researchers did was imagine different coding schemes that an extraterrestrial intelligence might use to code information in these 121 codons. And so they basically made up different codecs, 1 bit per codon, depending on how you like. And if you go into page 3, or excuse me, figure 3 of their paper, you can see they made 11 by 11 matrices to develop patterns, assigning different bit schemes to the codons, and what do you get? So figure 3A in the top left basically looks like a weird game of Tetris, and then the other figures with the different coding schemes don't seem to reveal the alien message, which is what they concluded. They could not find a message from an extraterrestrial intelligence in the phi-X genome, as humans isolated it from nature. Nevertheless, you can ask questions like, could you code for interesting patterns? What did we expect to find? A smiley face or something else, or could we make that? Now, in the context of synthetic biology, let me try and turn the corner here. When you return to biological systems and don't just look at them, but start to tinker with them systematically, you begin to uncover things that are a little bit beyond what are typically represented in our textbooks. So for example, here's a different map of the phi-X genome. This is unpublished work from Paul Jaska, a postdoc in the lab here at Stanford. On the outside of this set of circles, if you will, these arcs, are in black the original charismatic 11 genes that were identified in the genetic screens from Clyde Hutchison and others. But inside, you see many other putative open reading frames. And in fact, the number is quite bigger than what's shown on this figure. We can find these more easily now by doing comparative bioinformatics across many sequences of phi-X-like phage, and look for open reading frames that are at least 60 nucleotides long, that have strong putative translation signals, and so on and so forth. And this is quite typical. Most natural genetic systems have layers of complexity beyond what we typically celebrate. Now, nobody's found strong functions or phenotypes associated with each of these, so who knows? But what we can do now is use synthesis of DNA, and we published a paper on this late last year, to rebuild variants of the phi-X164 genome and see what happens when you change the genome architecture. For example, in this figure here on the bottom, you can see we took the natural phi-X genome, now shown as just a horizontal bar, with the overlapping genes, B inside A, K and A overlapping, K and C overlapping, so on, and we unstuffed it or decompressed it, as shown on the very bottom. We call this refactoring of a genome, where each gene now gets its own genetic real estate. When we put this unpacked genome into the cell, we found that phage is fine. It would seem to plaque and plate and lyse cultures just as well. What we've since been doing is going in and erasing all of the putative cryptic genes to see if they matter. And so now we've changed about 2% of the phi-X genome, erasing 100 cryptic open reading frames using synthesis to erase them all simultaneously. And what we're finding is there's a significant fitness drop-off, about a 90% drop-off. Don, in the control room, I don't know if you can hear me, but one question came in asking for the volume to be turned up. So I don't know if you can handle that, please. In any case, synthesis of DNA allows us to approach genetics via reverse genetics, making many, many changes simultaneously and seeing what happens as a result. So it's an approach to a complementary approach to scientific discovery. You can imagine sequencing a genome, annotating the genome, and then resynthesizing the genome, only including the things that you annotated to see what you missed, basically, as a direct affirmative test of your knowledge. I'm going to go on, call that an appetizer. I want to go on and talk about computers. And again, this is in the context of synthetic biology. So to ease into this, here's a nice photo. So let's all just relax a little bit. I'd like you to think about if you had to make a computer from the materials available to you in this photograph. Today, if you had to make a computer, what materials would you take? Would you take the fronds from the palm tree? Would you take the silica in the sand? Would you take the water? Something in the clouds? Maybe there's cloud computing, really? I don't know. In any case, well, we know how to make computing systems with silica. So let's start with that for just a moment. You can't make a computer directly from the sand on the beach, at least not a computer that's very powerful. We have to take those materials and refine them, process them, organize them into incredibly sophisticated objects that carry out the computing that allow us to have this web-based conference and many other things. Imagine taking biological materials and putting them through the same magnitude of transformation, refinement, standardization, processing, and so on and so forth. Could we get to a new type of computer that would enable us to do computing in new ways or in new places? And so in this next part of the talk, what I'd like to walk you through is building computers from not the silica shown in this photograph, but from the bacteriophage, which are in this photograph, but you can't see them because they're so invisible. They're all throughout the water in the oceans, for example. But the meta point I'd like to get across is that part of synthetic biology isn't just about understanding or tweaking natural biological systems. It's beginning to systematically rework biological materials to create a synthesis of new types of objects that could be quite distinct from what we find in nature, although they might be manufactured from biomaterials. Yeah, so Victoria has got it exactly right. You can make part of the computer from the sand via the glass. Keep that in mind, but now imagine implementing new types of computers from process biology. So I'll introduce you to three people real quick in this part of the talk. Here are the first two. Tone in the middle is a PhD student from Thailand, and Jerome on the right is a postdoc from Montpellier, France. And these two gentlemen have implemented data storage and logic inside cells using components from bacteriophage. The first part of this story was published about a year ago, as shown below, and it's building on ideas from the 1980s since. The idea here is if you wanted to encode 1 bit of digital information inside a cell, how could you do it? And Jerome's idea was to use double-stranded DNA within a chromosome as a data register. And the neat thing about double-stranded DNA is because you've got the 5' to 3' directionality of the backbone of DNA, you can basically have DNA in one of two orientations, pointing in one direction or pointing in the other direction. And that's it. As a molecule, the orientation of a DNA sequence within the context of a larger DNA fragment is perfectly digital at the single molecule level. Now that's obvious when you think about it, but you got to think about it. So Jerome calls this thing the flippy. And in this picture right here, it's pointing to the right. Right? If we could flip it with an enzyme, which we're going to take from bacteriophage, we'll call it an integrase, as that's what it's called, we give a pulsive expression of the integrase, and that'll flip the flippy. Now it's pointing left. And you can see here, for example, if we bracket the flippy with other genes, for example, a green protein or a red protein, we can indicate what the state of the flippy is by just causing those genes to be read out. A green cell would mean that the flippy is in state 0. A red cell would mean that the flippy is in state 1. So for example, we can take cells in the green state, that's shown in the microscope image at the bottom. The middle row here shows two-color cytometry. If you see a cluster of cells towards the bottom right of the graph, that means they're green. If a cluster of cells is in the top left of the graph, that means they're red. So the cells all start out in the green state, and we provide a pulsive expression of integrase, and the flippy flips, and now it no longer expresses green, it makes red. That means it's gone from 0 to 1. Then importantly, when we remove the input signal, we no longer express integrase. The cells stay red. So they're recording a memory. This is a non-volatile memory in the context of memory architecture. It means we don't need to keep gene expression on in order to maintain the state of our data system. So that's setting. To flip it back, you need to take the integrase and reverse its directionality by providing another protein cofactor, the excisionase, and that's going to cause the flippy, in theory, to go from the left-facing state to the right-facing state. And now we've completed a cycle if we can do this. This has been demonstrated as possible by Graham Hatfield's lab in a test tube, but it wasn't obvious how to do this in a cell. So for example, although I don't have all the data to present right here, you can see it in the paper. The first several hundred times we tried to reset one of these flippies, instead of getting the cells to go back from red to green, we got the cells to go back from red to yellow at an intermediate state where there was just bidirectional flipping over and over and over and over again. And that was because we couldn't figure out the right ratio basically to support the driving of the reaction backwards, the right ratio of excisionase to integrase, and then realize that ratio inside a cell. What I'm showing on this slide, though, is one of our better outcomes, where the cells all reset back. But you can see that when you remove the reset signal and the proteins go away, it looks like one of those cells that reset changes its mind and goes back to being red. And you can begin to imagine why that might be the case. For example, if in that cell the excisionase protein degraded more quickly than the integrase, then following a successful reset, in the afterglow of that reset, you might be in a situation where that cell has integrase all by itself because the excisionase degraded more quickly. That means you now have a set environment and you get a spontaneous corruption of the data. It took us a long time to do this, and we don't have time this morning to go through this entire table, but basically what this table shows is when we tried to implement the set and tried to implement the reset, and then tried to implement the set and reset in the same cell, all the different types of failure we got, from spontaneous flipping to no flipping to bidirectional switching to corruption of the set via fixing the reset and vice versa. And altogether, basically what this turned out to be was 3 years of work and 700 to 750 DNA designs, different design attempts to get 3 enzymes under control, such that we could get reliable set and reset. And that's what's shown here. We were finally able to do this, but you could go from cells in one state, looking at the top row here, just with one of the 2 colors shown on the y-axis, cells in state 1, then they're reset to state 0, they hold in state 0, set, reset, reset. This is the first time anybody was showing cyclical, multi-cycle, non-volatile, rewritable data storage inside a chromosome. So it's a great result, except for somebody like Jerome, who thought he was coming to a hot bioengineering lab and he'd be able to take standard biological parts off the shelf and get everything to work right away. Now, that just wasn't true at all when he started this project in 2008. It took him 3 years and 700 attempts. And the reasons for this were twofold as we understand it. The first was, at the start of the project, we were ignorant with respect to what levels of enzymes we needed to make precisely in order to get good switching. But later on, as the project developed and we gained that sense, we didn't have the tools, whether it was computer modeling software or parts off the shelf for controlling gene expression, that just worked. And so we basically ended up having to screen our DNA designs to eventually find something that worked as an integrated system. So that was the state of synthetic biology, if you will, or genetic engineering even, just a few years ago. Okay, that's data storage. Let me go on to logic and communication in the context of computing. And I'll come back to how easy or difficult it's been to get these systems to work, because it's changed quite remarkably in the last 12 months. A couple notes about this data storage system. A lot of times when you think about toggle switches or genetic data storage, as it's been practiced in synthetic biology, people have used cross-regulating transcription factors typically. And those are systems where when the system is in state 1, it actively represses state 0 and vice versa. So that's mathematically called a bistable system. The system we're making has no feedback. There's no cross-regulation between the states. And so it's not formally bistable. It's very interesting. Nevertheless, it's incredibly stable. So SSR here means the spontaneous switching rate. When we grow these systems out to 100 generations, they don't spontaneously change their state once it's set. It'd be like a light switch spontaneously going from off to on or vice versa. And we didn't understand this until after we published the paper, basically. And in trying to think about how it was working, we realized that what we had made was not a formal bistable switch, but instead what an engineer would call analog to digital converters. Now, what I mean by that in this context is we have an analog signal as an input. This is going to be the level of transcription from 0 to maximum transcription along DNA. That's a continuous variable from low to high. And then at some threshold of that analog signal, we either flip the switch or we don't flip it, right? So switch is not flipped, the analog signal comes up, we flip, or we don't flip. So we have a digital output. And as soon as we had A to D converters, we tried to black box them and ask ourselves, what else could you make with analog to digital converters or A to D switches? And in thinking about that, we went back to the work of George Boole, circa 1854, when he was analyzing human language and how we represent the world that we encounter. And so if you've not read Boole's investigation of the laws of thought, I haven't read the whole thing, and I'm not a scholar of Boole, but I appreciate it. And I point you to this paragraph here, where he's basically bootstrapping from language analysis the fact that we human beings have found it useful to observe the universe and make sense of things by using words such as and, or, and so on. And so he writes here things like barren mountains or fertile vales are useful examples of the word or. Um, or he also writes, men and women is the same as women and men, and so on. It's very interesting. We simply find it useful to have these logical operations for representing the world. In fact, we've found it so useful since Bull wrote this down that we basically implemented logical operations in every type of system we can engineer. So for example, Claude Shannon, shown here, worked out how Boole's logic might be mapped into electrical relays. If you look at table 2, I think from his, I can't remember his table 2 or table 1, from his master's thesis, he's basically working out how you could implement Boolean logic in logical relays. We've worked out collectively as a civilization how to implement Boolean logic in mechanical systems, in hydraulic systems, in electrical systems, and, well, now biological systems, as you'll see. The reason I give you this background is a lot of times when I share the work that's just coming on the next few slides, a scientist, a biologist might correctly say, biological systems don't implement logic that way. And that could be true, and that's a fine observation, and I don't care. The reason we're going to implement logic that we can control inside biology is so that we have logic we control inside biology, because it will be useful. So, to unpack how this was done in a type of an electrical system that inspired how we set up our own approaches. This is the transistor, a 3-terminal device that controls a switch which regulates the flow of current through a wire, shown as an original prototype schematically, and then in the paving stones of a university in Portugal, and so on. So many people know about the transistor. It's promulgated worldwide, and has been quite useful. We're going to implement something inspired by the transistor, but different, not a transistor. We call it a transcriptor. And it's going to leverage the fact that we now can flip DNA because we have really nice analog to digital converters. So the way this works is instead of flipping an element that turns on gene expression, we're going to flip an element that stops gene expression, a so-called transcription terminator. And the way this works is you have a flux, a current along DNA. It's not electrons on a wire. It's RNA polymerase, the molecule that reads DNA, flowing along DNA from left to right. And you're going to have to use your imagination. Usually when I present this, I've got nice animations, and you can see a YouTube video on our lab's webpage if you want to see this with live animation. But the way this works is if the transcriptor is in this orientation, the RNA polymerase is stopped at the T inside the transcriptor. But if we can flip that terminator upside down, it's going to be a unidirectional terminator, and that will mean that the polymerase can flow through. Hector's got a good question. I'm going to come back to it when we get to the bigger discussion around, is it either or building an airplane with staples? So that'll be a fun thing to talk about. So for example, if we put 2 transcriptors in series, each regulated by its own integrase that flips the terminators, this will allow us to implement a Boolean AND gate, right? So if neither terminator is flipped, if neither transcriptor is switched, the RNA polymerase is blocked, and nothing can flow to the right. Right. If we could flip them, or if we start it in a different configuration, you get pass-through all the way. You get a high output signal to the right. If it starts in this configuration, it's actually a different gate. It's called a not-or gate. Here's a different example. This is where we're nesting the flipping sites around a single transcriptor. So in this configuration, there's no polymerase flowing through. Neither control signal has come in to flip. If one of the control signals come in, then the polymerase can pass through. But if the other control signal comes in, then the transcriptor is returned to its original state, and the control signals have now turned off the output. So this is called an exclusive OR gate, where the output is high, transcription goes through only if the input signals are different. And I'm showing down below a schematic for how an electrical engineer would implement an XOR gate. It's actually quite complicated. You have to create a layer of 4 not AND gates. It turns out that in genetics, we can do something that's much more efficient architecturally than in electronics. And what's going on here is instead of having transistors where there's only one control signal regulating a switch, because the integrases are self-mixing and binding whatever site they recognize on the DNA, we can actually integrate multiple control signals within the same switch. And so that allows us to do a second order architecture that's much more efficient, that's better than electronics. I know that's a little bit esoteric, but so it is. Basically then, zooming out and being abstract for a moment, what we've implemented is a three-terminal device where we have a transcription signal as an input, we have a transcription signal as an output. Where is the input signal coming from? I don't care. It's anything that sources RNA polymerase. Where is the output signal going? What's it going to express? Anything that can be transcribed. Again, I don't care. As an engineer, we're designing these things to be agnostic to the input and output. The control signals come in as a third signal, regulate the expression of the recombinases that cause the transcriptor elements to switch or not. And there you have it. So that's the third control signal. By slotting in different logic elements, the 6 ones shown below, into the little gap, just by changing the logic element, you can get a different logical function. If you want to read more about what we mean by a transcription signal or the flow of RNA polymerase, formally we call this polymerase per second, or POPs. We use this as what an engineer would call a common signal carrier. We're doing this so that our devices can be connected up to any other device that uses the same signal carrier. So if you were a biochemist studying a promoter, you might think of a promoter as having a promoter clearance rate. POPs is kind of like that, but it's now formalized to account for things beyond promoters. And there's a free comic book online that discusses this, and you can just find it by searching for Adventures in Synthetic Biology or what have you. So we built all these systems. You can see the logic gates on the left, the truth tables for how their behavior should be. In the next column, the logic element architecture. In the predicted column, this is our modeling of how the behavior should be as the control signals 1 and 2 on the X and Y axis go from 0 to 1. The color indicates the expected behavior of the gate, blue being a 0 or a low output signal, red being a high or true output signal. So you can see the AND gate is high only in the top right of the predicted plot. Both control signal 1 and control signal 2 need to be high in order for the output to be predicted as high. And so it is down that predicted column. We built these, we put them into cells, we tested them. The measured column shows bulk fluorescent data from a culture of cells. The right column shows single cell distributions from cytometry. When we made these systems, every DNA design worked the first time. It wasn't 3 years of work with 700 design attempts. It turns out that we had actually successfully abstracted analog to digital converters that could be reused off the shelf. We knew enough about designing the logic elements with the recombinase recognition sites that worked off the shelf. And so it took us 3 months to do this work and 3 months to publish it. A very different experience for us, leveraging some early returns in a decade of synthetic biology research that was wonderful, but now we're sort of thinking about what to do next, basically. Just go into a little bit of detail on this to give you a sense of why this might be interesting from a control perspective and a logic perspective. These are 2 values in a switching experiment showing how the control signal in blue, as the control signal changes from low to high, the control signal is the thing regulating switching. How does the gate output change? So the gate output is in red, the control signal is in blue. These are contour plots showing the distribution of cells in a cytometry run. So it's a distribution of single cells. The darker solid contour, that represents 50% of the cell population. So half of the cells are inside that darker solid contour in both the blue and red case. So you can see on the left side of this plot, the control signals, averaging around 10 to 20 units, and the gate output is averaging around 50 units. We start to slowly increase the control signal. It doesn't look like the blue average is going up, but the contours are changing, the distribution changing, so the average is going up slightly. By the time you get to the 4th entry, the 4th from the left, you see that the control signal has gone up by about a factor of 5, but the gate has switched. The gate has gone up from an original value of about 70 to now an average value of about 7 or 8,000. So we've got about 100-fold switching in the output in response to just, it looks like about a 5-fold switching in the control signal. In other words, a very modest change in the control signal is sufficient to flip the switch, and that allows us to drive a very large change in the gate output. And the reason we can do this, among others, is we're using enzymatic unidirectional processing of the DNA, right? So that allows us to get really nice switching, but we're also using a third signal that independently is regulating flux along DNA of the RNA polymerase on a different molecule of DNA. And so that's going to allow us to go from a minimum to a maximum transcription rate with an enzymatic process. Again, I know that might be a little bit esoteric, but this was the first time folks had shown, in the context of engineered genetic logic, amplification of the control signals, which is significant because if you want to combine logic elements in series, you need to have a big enough output range to drive the next gate when you daisy chain them together. Okay. Last bit on computing. This is Monica Ortiz. She just finished up her PhD from Stanford and is heading to do a postdoc with Radhika Nagpal in engineering at Harvard. Hang on just a sec. Yeah, if there are multiple signals, could they interfere with amplification? It depends on, so hang on just a sec. Let me see if I can take this question. This is from Alitza Boteva. Thanks for that question. If there are multiple signals, could they interfere each like amplification? It really comes down to the integrases and how they recognize DNA. So we have Jerome, thinking ahead a little bit, didn't just take any recombinase to use in our systems. He down selected for serine integrases, basically, of the type discovered and characterized by Graham Hatfull at Pittsburgh or Maggie Smith in the United Kingdom. The down selection to serine integrases are because they're known to be unidirectional as opposed to bidirectional, and they also seem to not require host cofactors. So they might be isolated from a phage that infects a particular organism, but we could move them into yeast or E. coli or a newt or whatever. So we're down selecting for unidirectionality and portability. Having said that, those are stringent filters, and practically we only have 4 integrases in our laboratory today that recognize different sequences on the DNA. So the place where you get into trouble is if the multiple control signals power up the expression of integrases that then crosstalk with respect to how they process the DNA. So hopefully, Alitza, that's helpful. Let me jump back to Hector Zanil's question. So he writes, it's not whether biology works. So this is when I was trying to sort of provide some context around when we go to implement engineered logic in a living system, is it like biology or not, or whatnot? So let me just take this question out loud. And it's not whether biology works or not, one way or another. Thinking of logical gates in biology sounds like trying to build a plane with staples. How scalable is it to put logical gates together to create anything reasonably complex without giving up the controllability that you want to force by thinking in logical gates? I've got to think, Hector, a lot more about what it would be like to build a plane with staples. So that's an interesting metaphor. The way we think about it, the way I think about implementing logical systems in biology is I'm not trying to replace silicon. I'm not trying to replace mechanical systems. I'm not trying to replace any other type of logic. I'm trying to get a little bit of logic that we can control, you and me, everybody, in places where other types of logic don't work. So there's absolutely no chance that I'm going to be able to put silicon-based logic inside every cell of my body anytime soon, or even in the microbes in my gut. But a little bit of logic that I control genetically, maybe I could. So for example, after we published the logic work, just a few months ago, we had people in the medical school at Stanford who work with nautobiotic mice, where they study how mice colonize the gut. And they're very curious how the microbes the microbes colonize the gut of the mice. They're very curious how these microbes respond to signals from the mammalian immune system, and also how they respond to signals coming through the food supply. And so, typically what they do is they have expression markers for different responses of these cells as they're exposed to different microenvironments within the gut. And they'll either sacrifice the mice or, well, basically that's what they do, and then stain and try and figure out what the microbes experience. Now what they've done instead is they've taken our very modest logic elements and have them be responsive to different types of signals like fructose or an immune signal, and then record, did the microbe experience an environment where fructose was high and oxygen was low or something else? That triggers a flipping in the DNA. They can then collect those microbes in the feces and do a PCR assay or a high throughput sequencing assay and get a non-invasive measure of what was going on at a particular microenvironment in the alimentary canal. Nevertheless, I'll come back and sleep on the idea of planes with staples, and maybe we can follow up on that later. Okay, we've got 23 and a half minutes to go, so let me push on. Monica here worked on cell-cell communication. That's another part of a computer. She's adapting phage to do this. In synthetic biology, the early work on programmed cell-cell communication is obviously motivated by it's nice to engineer individual cells, but many things in biology are realized through ensembles of cells. How do we engineer many cells together? The early work in doing this, pioneered in Tom Knight's lab by Ron Weiss, was to adapt Bonnie Bassler's and others' work on quorum sensing in bacteria, where bacteria send messages back and forth via modified sugars, the homoserine lactones, for example. This is all great, but the limitation of messaging with sugars is the message you send is typically the sugar. And so it'd be like having a telephone, and the only thing I can say into the phone is sugar, sugar. And I can change how quickly I say it or how slowly I say it or how I can say sugar, or I can say sugar, but that's all I can say. I can say one thing. We're familiar with communication systems where the message we transmit through the channel is not determined by the channel for communication. So I could say, hi, Mom, how are you doing today? Or, dear funding agency, could you please send me more money? Or whatever I want to communicate through this channel. Could we do something like that in coordinating behavior among populations of cells? And the answer is yes, and it's pretty straightforward. Instead of sending specific molecules back and forth that are predetermined and not easy to change, how about sending DNA back and forth or enabling cells to autonomously transmit arbitrary DNA messages among themselves? And so here's the paper Monica published late last year. You can take a look at it, but I'll give you the highlight. Basically what she's doing is she's repurposing phage M13, which is a filamentous phage that doesn't lyse cells. It's secreted from the cell to package arbitrary DNA sequences autonomously and transmit them among cells. So for example, on this slide here, you see a 10-centimeter petri dish. On the right side of the petri dish are cells which are red, producing a red message. On the left side of the petri dish are cells that are green, producing a green message. These cells are modal. Over a period of a day or so, they swim towards the center, they collide at this wavefront, and they try and communicate with each other by sending their color in this example. If you pick cells right from that interface and you don't regrow them and you just immediately look at them under the microscope, that's what the top image shows here. You can see there's some red cells, some green cells in proximity, and about 30% of the cells turn out to be yellow. They've communicated their DNA. If you do this not in a semi-solid media format, but in liquid that's well mixed, you get about a 95% messaging rate in 3 hours without any genetic selections. If you do genetic selection, as shown in the bottom, you bring your message receipt rate up to about 100%. Okay, so now we can send any DNA message we want between cells. Oh, guess what? Our logic gates are made in DNA, so we can send them back and forth, and we've done that, and that works. So now you can begin to basically coordinate behavior over time and across space by using logical operations to program bacteria or other types of cells to behave in different ways. The messaging package we're using is a phage from bacteria, M13, but people have modified the M13 particle to enable it to transduce mammalian cells and other types of cells. So we imagine, we haven't done this, but we imagine that somebody could pretty readily take our DNA messaging system and have it be capable of doing trans-kingdom messaging from microbes to mammalian cells quite directly. Okay. So I wanna in maybe the last 10 minutes before we go to more Q&A, try and get to the engineering biology and give you some perspectives on synthetic biology. So this is something I'd like to make. It's a static image, but you can see there are 8 registers here pointing in different directions. Some of them are flipping. This is something I'd like to make. This would be an 8-bit combinatorial counter that could count up to 2 to the 8th. And for me, I'd like to have something like this operating on a chromosome and being responsive to cell division events, right? So I could count how old a cell is and then use that to tell me directly through combinations of fluorescent proteins the age of a cell as a tool for studying aging, studying development, maybe reprogramming development. How am I possibly ever going to engineer something this complicated? Could we get a lot better at engineering biology? Well, for context, 10 years ago, we published a report that was the result of an 18-month study where we were tasked with the question, how could you make biology easier to engineer? And the slides from this talk will be available. You can see that DOI number down below. That's a free download if you want to take a look at the report. This was commissioned by a funding agency in the United States, and we briefed the director of the agency 10 years ago, and they didn't like our briefing at all, so nothing came of this proposal. But the charge to the group was, how could you make routine the creation of synthetic biological systems that behave as expected? The DNA designs always work the first time. And the conclusions we wrote down a decade ago were do 3 things. Standardize biological parts, develop abstraction as a way for managing biocomplexity, and use DNA synthesis to separate the design of DNA from the fabrication of genetic material. Another way of thinking about the conclusions is at the sort of core of engineering, there's a design build test cycle. We would like to go around the cycle more quickly, more smartly, faster, more times in parallel. We're agnostic with how to get better, but just let's get better. And DNA synthesis might help with separating design and build, allowing for specialization. Standards for measurement and reuse of parts might power up testing and design. Abstraction for managing complexity could help with design. If you've never heard about these things before, here's the 1 minute on each of them. DNA synthesis, the chemistry is perfected circa 1982, the so-called phosphoramidite chemistry. It allows you to take chemicals in these bottles, the phosphoramidites, put them into a machine and have that machine be programmed by information coming in off a computer and print genetic material from information. The biggest thing printed in 2004 was 10,000 base pairs. The biggest thing in 2010 was a megabase. Another 100-fold improvement would get us to 100 megabases this decade. That's quite a large fragment of DNA. What would you do if you could print 100 megabases of DNA? Well, it obviously impacts the engineering process because you don't have to spend so much time with genetic engineering. You can just print your DNA. So for example, in 2005, when we redesigned the T7 genome, as shown in this paper, this is what we had to do. This is the first paragraph of the methods section where we used genetic engineering tools to manually engineer a 12,000 base pair fragment of DNA. This paragraph represents 3.5 person years of work by the best people on my team at the time. DNA synthesis replaces this work with this sentence. Please make this DNA for me. So there's a huge transition in the workflow for engineering biological systems. Another neat thing about DNA synthesis is when you combine it with DNA sequencing, it allows us to complement the top sketch here, which shows a biological system in a lineage passing from one generation to the next via direct descent, with the bottom process. So you can take a generation at a particular point in the lineage, sequence the genetic material, thereby converting some aspects of that organism into literal information. That information can be transmitted over time and across location. It can be resynthesized to the extent we're able to do that. And if we can take that material and reinstantiate a replicating lineage, off it goes again. So the significance of sequencing and synthesis together is they make genetic material and genetic information interconvertible or fungible. And that's going to allow us to decouple the promulgation of living systems from the constraint of direct descent. That's all very abstract. Let me give you a fanciful, fantastic, meaning it's not true yet, example. And it may never be true, but it's something to help you think about it. So the object on the right here, this perfume, is currently manufactured in centralized facilities and then shipped as material to people who would like to smell in a particular way. Imagine instead if we had a centralized research facility that was figuring out the DNA sequences encoding the biosynthetic pathways that made the odorants represented in the perfume. Those DNA sequences were then uploaded onto the internet, perhaps into iTunes. People all over the world could download them for 99 cents. They could use their local DNA synthesizers to synthesize the DNA. They could take that DNA, transform their epidermal ecosystems, the microbes living on the skin, and make living programmable perfumes. Right? That's an example of how the interconvertibility of sequence information and sequence material could change a lot of things. Standards. You've never heard about standards? No problem. They're all over the place in our society, screw threads, whatnot. Here's a nice example of standards from Segovia, Spain, where people assembled this aqueduct, not from natural rocks, but from standard rocks. They did this 2,000 years ago. And the significance of standards is that they allow for coordination of labor among human beings. So work can be coordinated across different locations or over time. If we can figure out how to coordinate labor or cooperate better, we can make things that individually would be impossible to realize. In synthetic biology, there are 4 types of technical standards, 4 categories being developed. One is just how to lay things out on DNA. That then supports functional composition. How do you design things so that when you put them together, they actually behave as expected? The third area is how you make measurements, so-called the science of measurement, metrology, reference materials, and so on and so forth. You can think of the meter stick as a reference object. What are the units for gene expression? How would we compare them directly? The Miller unit doesn't cut it. The fourth area of standards is basically data exchange, representations. How do we describe things and share that information? This has been very controversial within the research community. So for example, for many good reasons, people have observed that you're never going to be able to make standard biological parts because the functions of molecules always change as the environment changes, or everything inside a cell is connected to everything else, and it's just never going to work. Could be, but the data is starting to come in. So for example, from the BioFab project, Vivek Mutalik led a team where he was able to take translation initiation elements in prokaryotes, which are particularly difficult to reuse because of the folding of the RNA across the junction between a ribosome binding site and the coding sequence, and actually demonstrate a novel genetic architecture inspired directly by nature that supports reuse of these parts. And he's now made an incredible free-to-use collection of standard biological parts that can reliably express proteins with high precision, independent of the coding sequence of your gene of interest. So when we implemented our logic gates in every DNA design work the first time, we took advantage of these very recently published parts collections to get better composition and better behavior. So we believe this now to be possible. For example, in the context of abstraction, we'd imagine some future where somebody could lay out very complicated high-order devices like an 8-bit synchronized counter, but instead of working down at the DNA level directly, they would call down to somebody working on a data latch and say, please give me 8 data latches that I can put together. The person working at the top level, if we can implement this type of abstraction hierarchy, might not need to know that DNA is made up of 4 bases. Kind of like we're having this talk over the web right now, and I kind of know that the computers are running on zeros and ones, but if you ask me to program a computer in machine language, I can't do it, right? Because there's a functional hierarchy that compiles down to the computing that insulates me from that. When we did our data storage project, we were struggling to go between the data latch layer in the middle and the parts layer down below. We had to go between those 2 layers 750 times because we were ignorant of what we needed to make in terms of levels of expression, but also because we didn't have the parts. When we made the logic system, we had the parts and knew what we wanted, and so we only had to go across those 2 layers once for each DNA design. So that's a type of example of abstraction whereby black boxing analog to digital converters and having reusable parts, we gained a huge efficiency in engineering stuff. That isn't to say that this is all done. It's simply now the case that after 10 years of sputtering work, we've shown that these basic ideas from 2003 I would represent are not impossible to make true, and that when you make them true, they're hugely powerful. So to try and put this in context, one of the critiques of synthetic biology is the same old thing as genetic engineering. I don't view that as being correct. I view synthetic biology as a second generation of genetic engineering, an outgrowth. And to sort of lay that out for you, if you'd like to think about it, and maybe disagree or agree. Across the top row here, you see the core tools underlying genetic engineering work. Coming in the bottom are a new category of tools that should keep going. I expect more to arise that allow new ways of empowering the design, build, test, learn cycle in engineering biological systems. Hector continues to represent that he doesn't think Boolean logic will scale in biology. Maybe, maybe not. I think we're showing that it's going to scale already. We only need to get out to about 8 bits to do incredible things. We don't need to get to a gigabyte. 1 byte of data inside cells allows you to do things like count to 256. If you look at the depth of developmental lineages, if you look at aging, counting that many cell division events, counting hundreds of cell division events, covers most of what you want to study or control. So we don't have to push the scaling too far to basically win. I'll wrap up with just a couple slides and maybe jump to the end. As this work has progressed, it turns out to ramify in ways that aren't just technical, they're cultural. So standards allow coordination of labor. Here's some of the first classes we taught. Actually, this is the first class we taught 10 years ago. These students were sharing and reusing parts as best they could. And so they kept doing that over the years, and more students started to join them and started to get projects working and publishing research papers. And it became known as iGEM, the International Genetically Engineered Machines Competition. And it got a little bit bigger over the years. China joined in 2006, for example, and they won the competition, got a little bit bigger, and it got a little bit bigger, and got a little bit bigger. And I can't show you photos from 11, 12, 13 because it just got bigger than the room. These students are taking the emerging tools of synthetic biology and more quickly working to address needs and opportunities that might be solved with biotechnology. Right? So in the United States, for example, 30, 40 years into biotechnology, we have a subsector of our economy that's about $300 billion a year of products realized through genetic engineering. That's about 2% of the US economy, which doesn't sound like a big number, but 2% of our economy is the same in the US as mining, extracting stuff from the crust of the earth. These students with new tools are now looking to define the future of the bioeconomy. So as an example, iGEM project in 2009 in England, with $25,000 in capital in 16 weeks, 8 teenagers engineered 7 biosynthetic pathways to implement 7 pigments shown here, what they called e-chromi, such that bacteria could produce different colors, depending on what was initiating the expression of the pigment manufacturing system. When they partnered with the designers at the Royal College of Art, the designers said, well, what do we want with colorful bacteria? They imagined a fanciful application where you could have a probiotic yogurt that would have genetic sensors responsive to the disease state of your gut or your alimentary canal, and depending on what illness you had developing, what came out of you would be a different color, thereby indicating what specialist you should go see. This sounds like a fanciful application, but note that most people can't afford to go to a doctor to get a diagnostic of their colon or gut or what have you. So a passive built-in biological diagnostic might be a fairly pressing application of biotechnology. As another example, we can look to new modes of biomanufacturing. So this is a pile of sawdust at a mushroom farm that uses wood fungus to grow mushrooms, as shown here. It's a totally incredible process. What if we could use wood fungus to grow other objects? This is the work of Phil Ross, where he's manufacturing chairs that grow themselves. This is a provocative museum piece that doesn't finish the chair, so it looks like it's still alive because it is. But you can actually take ground up plant waste and convert it into objects from food to materials to plastics and so on. We can do a lot more with that. I'm going to skip ahead just for the sake of time. Well, I apologize. I just have a few slides left, so let me try and finish, and then we'll have a few minutes for Q&A. If you haven't seen this before, here's another example of building with biology at a different scale. These are double-decker suspension bridges grown in place from the roots of a rubber tree. This manufacturing process fixes carbon over time. This is done in India where people are weaving together the root systems with, direction and sustaining the process over generations. So what we could manufacture with biology, I think, is well beyond the biotechnology we now experience. We just have to imagine it and enable it. I'll give you an odd, funny example, again, just to maybe make a memory for you. Um, we supported artists and biotechnologists working together to see what they would do with new tools. So for example, Sissel Talas from Berlin worked with Christina Agapakis at Harvard. Sissel is an artist, a perfumer, if you will, and Christina is a microbiologist. 1 month in the laboratory, 1 month in the art studio. When Sissel got to the microbiology lab, she asked what's exciting in microbiology, and Christina said the human microbiome, the microbes on our skin and in our gut. When Christina asked Sissel, what's exciting to you? She said the odors of cheese. So they went to the cheese store and they pulled core samples of the stinkiest cheeses they could find and brought them back to the laboratory to study the microbiome of the cheese. Initially, they were doing determinative microbiology, but pretty quickly, they started having questions about engineering and synthesis. So, these cheeses, some of them are made in very small batches by human beings manually who have skin and who have microbes on their skin. So they ask, what is the relationship between the human microbiome and the cheese microbiome? A scientific approach would be to sequence everything today and do the alignments and make some report. But a synthetic biology approach is a little bit different. They went around the Harvard campus and they collected skin microbe samples from 48 colleagues. And then they went to the farm and they got raw milk and they inoculated 48 bottles and made cheese from human skin microbes. So they made cheese from the microbes in Daisy's armpit, from the microbes found on the toe of a philosopher, from Christina's hand, from Sissel's nose, and so on. Now, the first time they did this, nobody thought about the biosafety, so we didn't taste the cheeses, but we smelled them. And I can report that Daisy's armpit cheese is beautiful. It's a citrus floral bouquet, whereas the cheese made from the microbes found on a philosopher's toe is wrong. You need a new adjective to describe how foul it is. This project was very interesting to me because it reversed what my mother had told me growing up. Eat your vegetables because you are what you eat. This project teaches me that we eat what we are to a degree. And so I think there's a hint within synthetic biology, not just with manufacturing, but with existence, that we might reframe the interfaces between the living world and ourselves by becoming a lot wiser. So as a last slide, I guess I just summarize this with a bit of dreams. By developing new tools, we're making biology easier to engineer. 10 years in, we've had incremental success at doing this. Who knows how far we can go with it? I think a byproduct is an amazing complementary approach to understanding life or scientific discovery. Opportunities include enabling all constructive biotechnology, not at some arbitrary point in the future, but this century. And if we do that right, we get to renew, not replace the jungle, but preserve it by renewing ourselves in partnership with nature. Thanks very much. And any questions, we've got a few minutes, I'm happy to take them. Thanks, Beverlyn. Okay, so I think we'll wrap it up there. If you want to follow up or look for anything, my email address is my last name, endy@stanford.edu. I'm happy to respond. If you do a Google search for our website, you'll find more materials there. Hope everybody has a great day and enjoys the rest of the presentations.